Is this on? Perfect. Well, it's 8 o'clock. So good morning, everyone. I'm Miklos Sehintat from UCLA, and my job is very simple. I'm just here to say welcome. And this is a historic meeting because the first time, I was going to say after the pandemic, but it's rather during the pandemic that we get to meet in person. And most of the crew is still having breakfast, and I hope they will be up. And traditionally, we open with a symposium, a pre-meeting symposium. And then this is usually the privilege of the president who picks the topic. And this year we have something very exciting about necrotizing pancreatitis. And without further ado, I ask Joe Hines and Ali Auch to start the first session.
Good morning, I'm Aliyah Uch.
Good morning. Our first session is on the biology of necrotizing pancreatitis. And our speaker is Dr. Anna Glukhovskaya from UCLA, and she's going to be addressing types of cell death and pancreatitis. She's a virtual speaker this morning. Skaya from UCLA.
Thank you for inviting me to present at this interesting meeting. And sorry I cannot be with you today. Because I opened the meeting, I will remind you some basic facts. Necrosis is a major complication of pancreatitis, but the molecular mechanism leading to acinar cell necrosis are not well understood, and approaches to reduce necrosis in pancreatitis are not established. Today, I will try to briefly summarize what is known about the different types of cell death in general and in pancreatitis, underlying mechanisms and approaches to reduce necrosis in pancreatitis. Necrosis can be caused by accidental mechanical, chemical, or thermal membrane damage, or by insufficient amount of ATP to maintain ion gradients through plasma and organellar membrane. Loss of ion gradients results in cell swelling, loss of membrane integrity, which is a major feature of necrosis, leakage of cellular content, which results in inflammation, in attracting of inflammatory cells. This cartoon is from the review by Douglas Green, who is authority in cell death field, and it is written as a play with different form of cell death as main characters. That's what he's saying about necrosis. I am cell death, I am disease. Apoptotic form of cell death is very different. Organism activates apoptosis to eliminate unwanted cells. Morphologically, apoptosis manifested by a cell shrinkage, nuclei, and then cell fragmentation into apoptosis bodies, which are phagocytosed by macrophages. Differently from necrosis, apoptotic cells maintain plasma membrane integrity. There is no leakage of cellular content and no inflammation. Apoptosis plays a major role in physiologic processes, in particular organism development, morphogenesis, as shown in this schematic. Apoptosis morphology was first described by Australian pathologist John Kerr, and he introduced the term apoptosis, which means falling cliffs from Greek, a little bit later. Nobel Prize in 2002 was awarded for identification of genes controlling apoptosis in C. elegans. The first gene was specific protease caspase. And this was what was Doug Green is saying about apoptosis. I am most of cell deaths that occur in a living animal. I am peaceful, powerful, and immunologically silent. I am a molecular disease. The predominant point of view in early 2000 was that non-regulated necrosis and apoptosis are the only types of cell death. However, later studies showed that in addition to non-regulated necrosis and apoptosis, cells possess multiple mechanisms of programmed necrosis. The major are necroptosis, pyroptosis, and ferroptosis. The same as non-regulated programmed necrosis is a lytic type of cell death. However, differently from non-regulated necrosis, programmed necrosis is controlled by specific genes. Thus, the current point of view that there are three major types of cell death: non-regulated necrosis, programmed necrosis, and apoptosis. The studies of acinar cells in death in pancreatitis started in late 80s, and remarkably, John Kerr laboratory was the first who described apoptosis morphology in acinar cells in pancreatitis. The subsequent studies investigated the role of cell death in pancreatitis severity, signaling pathway mediating acinar cell death and their regulation. Sorry, I only showed the first and the last author's name. The data indicate that acinar cell in human and experimental pancreatitis dies through necrosis and apoptosis. Both non-regulated and regulated necrosis cause rupture of plasma membrane, release of dumps, triggers of inflammation, and damage of neighboring cells. The severity of pancreatitis directly correlates with the amount of necrosis. Apoptosis, which does not stimulate inflammation and cell damage, is associated with milder disease. However, in pancreatitis model and in human disease, the percentage of necrosis is much greater than of apoptosis. So the question is, why apoptosis rate is low in pancreatitis? What are mechanisms of necrosis and apoptosis in pancreatitis? Abnormal calcium signaling is an early pathologic response of pancreatitis. Increase in cytosolic calcium causes mitochondrial calcium overload, followed by the opening of non-selective channels in mitochondrial membrane, then permeability transition pore. Its main component is cyclophilin D. PTP opening results in the loss of mitochondrial membrane potential, inhibition of ATP production, and ultimately necrosis. The type of non-regulated necrosis, termed PTP necrosis or PTP-mediated necrosis, is a major necrosis pathway in pancreatitis. Genetic ablation PTP with cyclophilin D, genetic blockade of PTP with cyclophilin D genetic ablation prevents depolarization, restores ATP level, and reduces necrosis. A critical event initiating apoptosis is the release from mitochondria its resident cyclophilin D. So in pancreatitis, both necrosis and apoptosis associated with mitochondria permeabilization. In cytosol stimulates formation of large protein complexes, complex called apoptosome, resulting in activation caspase 9 and then caspase 3, a major mediator of apoptosis. This pathway, called intrinsic, is a major mechanism of apoptosis in pancreatitis. Cells tightly control apoptosis using two major mechanisms. The first is BCL2 protein. The release of cytochrome C is regulated by 25 members of BCL2 family proteins, and the extent of cytochrome C release is determined by a balance between pro and anti-apoptotic BCL2 protein in mitochondria membrane. Cells also express and activate several inhibitory apoptosis proteins called EAPs, which directly block caspase activity. This schematic shows pathway of both necrosis, PTP necrosis to the left, and apoptosis. Pancreatitis also stimulates several anti-apoptotic mechanisms. Cytochrome C release in pancreatitis is inhibited by PTP-mediated depolarization and also by upregulation of anti-apoptotic BCL2 proteins. Caspase activity in pancreatitis is inhibited by PTP-mediated drop of ATP because caspase activation is ATP dependent. Also, but pancreatitis induce upregulation of inhibitory apoptosis protein. Thus, apoptosis in pancreatitis has little chance to prevail. This cartoon shows that program necrosis aids necrosis, PTP necrosis, to restrict apoptosis. However, little is known on the role of program necrosis in pancreatitis, as I will show on the last slides. The initiating event in necroptosis is complex formation between kinases RIP1 and RIP3, forming protein complex called necrosomes. This results in recruitment and phosphorylation pseudokinase MLKL, which forms oligomer, which insert into and permeabilize plasma and organelle membranes. Pyroptosis is a form of inflammatory cell death. It mostly occurs in macrophages, where inflammatory caspases cause proteolysis of protein called gasdermin D. Oligomers of gasdermin D, its fragments forms oligomer, which are embedded in plasma membrane, forming a pore. Finally, ferroptosis is caused by peroxidation of phospholipids in plasma membrane, also resulting in plasma membrane rupture. Thus, all three forms of program necrosis is lytic cell death. Necroptosis activation in pancreatitis is well established in studies which showed RIP activation, necrosome formation, and MLKL phosphorylation. We know much less about activation of other types of cell death in pancreatitis, of other types of program necrosis in pancreatitis. The recent report surprisingly suggests that inflammatory caspases cleave gasdermin in acinar cells, resulting in activation of pyroptosis in acinar cells in pancreatitis. These data, as well as some reports on ferroptosis activation in pancreatitis, require further confirmation. Even though necroptosis is activated in pancreatitis, its role remains unclear. Most of the studies found no effect of necroptosis blockade on cellular pancreatitis severity. One explanation is multifunctional role of RIP3 and RIP1. However, even genetic ablation of MLKL, which is specific mediator and marker of necroptosis, showed no effect of disease on disease severity, no effect on histology, necrosis, and even upregulation of inflammation. Definitely, the role of program necrosis in pancreatitis requires further investigation. In sum, current knowledge suggests that PTP necrosis is a major type of cell death in pancreatitis. Approaches to reduce PTP-mediated necrosis include inhibition of proteins mediating PTP opening, such as cyclophilin D and FIDAC, normalization of cell and mitochondria calcium signaling, which I'm sure Robert Sather will be talking about, enhancing autophagic efficiency with tricholos to eliminate damaged mitochondria. It is important to better understand the role of program necrosis in pancreatitis. The arsenal of chemicals to inhibit individual type of program necrosis is expanding rapidly. These inhibitors can be applied to selectively block particular type of cell death in pancreatitis. And these below just example of ongoing clinical trials. In the end, I want to show you a slide from a recent study by German scientist from Dresden, Andreas Linkermann, who shows clinical value of the detection of cell death pathway. There are biopsies of 2 patients with kidney disease stained for phosphorylated MLKL, a marker of necroptosis. You can see that patient A has a lot of positive staining and will be treated with necroptosis inhibitor, whereas patient B did not develop any necroptosis, and the treatment will be different. In the future, biopsies will be stained for the markers of various types of cell death, which would allow personalized treatment. So what we are to do? Simple, we only need to find out which pocket lady pushes the ball and how to redirect. Thank you.
Questions at the end. Thank you. Thank you, Dr. Gukovskaya. Just want to remind everyone that we will take the questions at the end. And also, I would like to thank the audience for wearing the mask at all times and above their noses if possible, unless you're eating and drinking. So it's very important for us to keep an infection-free conference. So that's how we can keep those conferences coming in the middle of the pandemic, as Miklos said. So our next speaker is also a virtual speaker, is Dr. Robert Sutton from the University of Liverpool. He's going to talk about the role of mitochondria and ischemia in necrotizing pancreatitis.
I'd like to thank Joe Hines, the president of the APA, and the program committee for the invitation to speak in this symposium. My name is Robert Sutton, and I'm going to talk about the role of mitochondria and ischemia in necrotizing pancreatitis. These are my conflicts of interest. The generation of adenosine triphosphate, ATP, is a principal function of mitochondria. ATP is the currency of energy in all living cells, and it is generated by mitochondria in the majority. Indeed, the majority is generated by mitochondria. Mitochondria also respond to calcium and other signals by upregulation of the citric acid and beta oxidation of fatty acid cycles. Mitochondria also respond to cell stresses, or indeed respond to the needs for cellular proliferation. When cell stresses become severe, mitochondria execute cell death pathways. The generation of ATP is conducted by a series of complexes located on the inner mitochondrial membrane. Complexes 1 to 4 generate an electrochemical gradient utilized by complex 5. Complex 5 is the F1 F0 ATP synthase, shown here as the F0 component, which is in the membrane, and the F1 component, which is in the matrix, joined together by central and peripheral stalks. In response to the proton gradient, the C ring acts as a rotor and then generates the ATP from a combination of adenosine diphosphate and phosphate moiety. Under conditions of cell stress, the mitochondrial permeability transition pore is activated. And there is increasing evidence that it is the F1 F0 ATP synthase that forms this pore. Most recent data suggest there is separation of the F1 and F0 components, allowing the C ring to expand, and that then forms a pore through which particles up to 1500 Daltons pass. And as a result, the mitochondrial membrane polarity is lost, the electrochemical gradient diminishes, and ATP production is inhibited. In the pancreas, there are several regions of mitochondria predominating. Particularly important is the perigranular region, which allows for stimulus secretion and stimulus metabolism coupling. Additionally, it protects the rest of the cell from all of the calcium signals spreading and allowing for the possibility of greater overload. Calcium signaling is crucial to stimulus secretion, as shown by Katsuhiko Mikoshiba's group. In acute pancreatitis, mitochondrial depolarization is a common feature as a result of calcium overload. This is seen in a toxin example on the left on this slide, and in a pressure example on the right in this slide. The top left and the top right panels show the calcium transients, very large, very prolonged, in response to these two stimuli. The middle left and middle right panels show the mitochondrial membrane potential. On the left, prevented by cyclophilin D knockout, the PPIF-, and on the right, prevented by pizzo-1 knockout. And the bottom panels on the left and the right show the histological scoring of acute pancreatitis. On the left, induced by TLCS, and on the right, induced by Yoda-1, a pizzo-1 agonist. And in the cyclophilin D knockout mouse, the TLCS acute pancreatitis is prevented. And on the right, the TRPV4 knockout mouse, the acute pancreatitis is prevented. So we see two different pathways actually acting through calcium overload and mitochondrial membrane depolarization. In the middle, we see the machinery, critical to which is the Mitochondrial Calcium Uniporter, or MCU. In health, responding to normal calcium signals by upregulation of energy utilization, and in calcium overload, allowing excess calcium into the mitochondria, which results in mitochondrial permeability transition pore opening. This is represented in the diagram here. The upper panel shows in health, generation of ATP with a closed mitochondrial permeability transition pore and normal functioning of complex 5 with a healthy cell and a healthy pancreas. In the bottom panel, we see a marked diminution of ATP production with opening of the MTTP in response to calcium overload. And as a result, hydrogen ions come in, the mitochondrial membrane potential is lost, ATP is not generated, the cells die by necrosis, and the result is acute necrotizing pancreatitis. Could it be that inhibition of mitochondrial calcium uniporter uptake prevents acute pancreatitis? Well, we can see here in the constitutive mitochondrial, in the induced mitochondrial calcium uniporter knockout mouse, the calcium signal is greater because there's no mitochondrial buffering. Calcium entry into the mitochondria is greatly inhibited, both in response to CCK and TLCS, and the reducing equivalent generation is also. However, there is no protection from acute pancreatitis. It could be, of course, that in future, should drugs be developed that can inhibit but don't prevent action of the mitochondrial calcium uniporter, we may find there is protection. The data thus far underline the importance of ATP generation and ATP in protection from calcium overload. In the left two plots, we see how patching in of ATP protects pancreatic acinar cells and allows them to handle calcium more effectively, restoring baseline calcium concentrations. And on the right, we see the impact of a high toxic concentration of TLCS, which induces large chloride inward current and huge calcium transients with death of the cells. Whereas in the cyclophilin D knockout, calcium is handled more effectively and there is restoration of basal levels and restoration of normal membrane potential with no cell death in response to excess taurolithocolate acid sulfate. What of the circulation? The pancreas has multiple macroscopic arterial collaterals, but the lobules are in the main supplied each by an end artery. Another important feature is the highly fenestrated capillary network, and both of these contribute to susceptibility to small vessel ischemic injury. Well, the pathological processes are legion: capillary leak syndrome, hypovolemia, hypotension, pancreatic hypoperfusion, hypoxia, hypercoagulability, and a greatly reduced functional capillary capacity. When that becomes severely restricted, necrosis ensues, and this is proportional to the severity of the disease. Mitochondria respond with hypoxia-inducible factor 1 and more anaerobic glycolysis, but this is often insufficient when the calcium overload is marked and the cell death pathways are triggered. In the presence of less ATP, necrosis is the end result. And with the microvascular abnormalities and mitochondrial compromise, amplification of these pathological sequelae occur. What are we to do about this? Well, the amplification loops in A and D may be addressed by Voxora, the calcimedica ori channel inhibitor, CM4620, previously CM128, same compound. And the phase 2b trial that they're conducting, we hope will be successful. The Goulash trial, undertaken by the Hungarian Pancreatic Study Group, we hope will address the diminished ATP production by early delivery of high quantities of energy via nutrition. And in addressing inflammation and the tissue injury that results and infective complications which ensue, the RAPID-1 trial of infliximab, we hope, will deliver results dependent on the EME program renewing funding. I'd like to thank the funders, and I'd like to particularly thank Anna Gukowskaya for work on the cyclophilin D knockout mouse, and for Roger Liddell for his work on the pizza-1 TRPV4 knockouts. Thank you.
Thank you, Dr. Sutton. Our next speaker is Dr. Stephen Pandal. Dr. Pandal is at Cedars-Sinai Medical Center, and he's going to be speaking on organ failure and pancreatitis.
Good morning, everyone. This is Steve Pandal. I'm so sorry I can't be at the meeting. I'm truly missing it this year. My talk for this symposium is on, excuse me, organ failure mechanisms in acute pancreatitis. And this slide also shows multiple of us who are involved in this study. They include Richard Waldron and Aurelia Lugea, who are in our pancreas research group at Cedars-Sinai in Los Angeles. Corey Bystrom, Sarah Parker, and Jenny Van Eyck, who are in our proteomics group at Cedars-Sinai. and George Papachristou, who is from Ohio State University. This slide shows that conceptually that organ failure plays a leading role in mortality and severe acute pancreatitis. And on this slide that was taken from a publication from Gamatos in 2014 shows that acute pancreatitis and early death and also potentially late death are associated with systemic inflammatory response and organ failure. In the late response, generally due to sepsis from pancreatic necrosis. For the information I'm going to show today, we use severity measurements that were proposed in the Atlanta classification system, first developed in 1992 and then revised in 2012. In this classification system, the patients are divided into 3 categories: mild severity, those with no organ failure; moderately severe category, where the organ failure in patients resolves within 48 hours; and severe pancreatitis, where there's persistent organ failure. I show this slide to give some context to what I'm going to be talking about. And it's from a publication by Rui et al. From 2020, where Dr. Bowen G was the senior investigator, and we collaborated with Bowen in this project. The important points about the project is that he developed an animal model of hereditary pancreatitis by inserting the human PRSS1 mutated human gene into the Asner cells of a mouse. What we found is that the Asner cells themselves, when we took them out and stressed them with stressors that cause pancreatitis in the test tube, basically we didn't see any difference from control Asner cells from wild type mice. However, in the whole animal, you see in the picture in the middle, there was increased trypsin activation, increases in the stress pathways, and more Asner damage that you see, Asner cell damage you see on the right, more inflammation, and even stellate cell activation, which is a precursor to developing more chronic forms of pancreatitis. The other thing that was observed was fibrin in the tissue, suggesting a coagulation disorder. And Bowen and his team actually had the idea of adding a prothrombin inhibitor, dabigatran, to see if that would improve the pancreatitis, and it actually did, which was quite an interesting observation in the field. So with that in mind, and also trying to understand better what the mediators of organ failure are, we developed the following hypothesis, and that is that detection of serum biomarkers of severe acute pancreatitis will reveal potentially novel mechanisms of organ failure. Our aims were to perform quantitative proteomic analysis on serum samples from patients with the different severity forms of pancreatitis: mild, moderately severe, and severe. And then to use the proteomic data to identify those that are differentially increased in patients with moderately severe and severe pancreatitis compared to the levels of those patients with mild pancreatitis. And this chart gives the demographics of the patients, and the samples were from Dr. Papachristou when he was at the University of Pittsburgh, and as I indicated earlier, has since moved to Ohio State University. And this gives the patient characteristics. We had 10 samples from each of the severity groups: control, mild acute, moderately acute, and severe acute pancreatitis. You'll see, looking through the chart, that we have some imbalance in some of the categories. For example, in the age groups, we have younger patients than we have with the different severities of pancreatitis groups. but we need to fix that with more samples of age-matched controls. The next characteristic of those samples is that we had different etiologies from this group of samples from Dr. Papachristou. And the graphic just shows that we had equal numbers of patients from control, mild acute, moderately acute, and severe acute pancreatitis. The different disease etiologies were gallstone, hypertriglyceridemia, ethanol abuse associated pancreatitis, and other forms. The proteomics data we developed is shown here, as well as the pipeline of analysis. And so we were able to determine the intensity or quantitative value for each of the proteins in the samples. We identified 533 proteins across the different severity groups. 221 were expressed in all of the sample types. We, in order to determine differences between the groups, we chose those proteins that were elevated. By the way, there were decreased proteins as well, but we used elevated proteins that were twofold in difference from control. And then we'll show you a Venn diagram of those. We also did various analytic methods to determine the upstream mechanisms that regulated those protein expressions. And they're listed here, like the David analysis and pathway analysis. So the instrumentation we used was an Orbitrap Fusion Lumos system for quantitative proteomic analysis, and it's a state-of-the-art instrument that you see on the right. And we used MaxQuant and Perseus for data analytics. On the left, you see that we analyzed serum, and that the serum was actually pre-treated before the sample was put onto the liquid chromatography mass spec machine. This is our first piece of data, and it shows the proteins that were differently expressed and increased in the blood of patients according to their acute pancreatitis severity level. And so you can see, for example, in blue, the mild had several proteins listed here that were increased in the mild severity category compared to control. The severe category had unique proteins that are in the Venn diagram, not crossing with the other groups that were only seen in the severe category that are listed in the yellow group. And that same is true for the moderately severe. There were a group of 4 proteins that were increased in that group uniquely compared to the other groups. And then you can see in the Venn diagram, there were multiple proteins that crossed between the groups. And on the right-hand side, you can even see that there were a total of 27 proteins that were common between mild and severe. The next step for us was to evaluate the proteins that were expressed in the severe and moderately severe groups compared to the mild. Again, those were chosen because those are associated with organ failure. We wanted to see if we could identify proteins that are increased in those patients that had organ failure that could help us understand better their genesis. So the next slide I'm going to show you is one of our first evaluations of the data, and it's by David analysis, and that shows proteins that fall into different kinds of pathologic pathways. And here we compared the severe and moderately severe acute pancreatitis compared to mild. You can see that the top pathways that show up have to do with the intrinsic and extrinsic prothrombin activation pathways, platelet degranulation, complement activation, blood coagulation, for example. I'm going to focus on those. And the next slide shows particular proteins that were increased in these different sets of pathologic pathways. I think it's important to see that FGG and F5 show up in several of the top pathways. So FGG is fibrinogen and F5 is factor V, which is interesting. We all remember those. And also there's some inflammatory mediators like CD14 that are important, probably in the pathogenesis related maybe to the endotoxin pathways. And this is just to show the blood coagulation cascade to just remind us of where factor V and fibrinogen sit. So factor V promotes the conversion of prothrombin into thrombin, and the fibrinogen was increased, so it creates a increased substrate for fibrin formation and clot formation. And also there were platelet degranulation factors that were identified as well that are on the right. This slide is another data analytic method called the Ingenuity pathway analysis. And what it tries to do is take the proteins that were, in our case, increased in the severe versus mild pancreatitis groups and try to see if there were some trunk pathways that could mediate the responses that we were seeing. And to point out again, FGG is here and fibrinogen activator is here. CD14 I pointed out before is on the left-hand side in this diagram. But importantly, in the center are potential mediators of pathways or systems that can mediate these responses. And you see pathways we certainly recognize like IL-6, STAT3, which is downstream of IL-6, AGT, which is also an inflammatory pathway system, and lipopolysaccharide that I referred to before. So these are increased much more in the severe pancreatitis. These proteins that I've represented are increased in the severe pancreatitis compared to mild, and those increases could be a downstream result of these central pathways that I just went over. So the last couple slides are just conceptual, and it's showing our concept that severe pancreatitis is associated with a coagulopathy, and that coagulopathy could be the mediator of organ failure, and at the bottom, cardiovascular, lungs, and renal failure. I also showed this slide at the end because we've been thinking that at least the pulmonary manifestations of severe acute pancreatitis could be similar to those of COVID. And some of the same pathways are being identified in severe COVID disease, like the thromboinflammatory and hypercoagulation states, and some of the inflammatory mediators that we've been discussing. So this just is for us an internal question whether some of the mechanisms are similar between severe acute pancreatitis and organ injury and what is seen in a severe COVID disease. Thank you very much, and I'll be open for questions in our discussion period.
Thank you, Dr. Pandol. And we will continue, and this time with a live speaker, Dr. Julia Meyerly, who needs no introduction. So she will talk about the local and systemic immune response to necrotizing pancreatitis.
Now it's working. Thank you very much. I wouldn't have thought that I would be the only live speaker in this session. However, it's a real pleasure to be again within this group, and I hope that we will have a lot of on-site discussions because I think we missed them all greatly in the last 2 years. So the topic which the organizers gave me today is the local and systemic immune response in necrotizing pancreatitis. So this is just to remind you, and I'm very aware of that I'm dragging oils to Athens. So pancreatitis starts in the acinar cells, and the damage to the acinar cells perpetuates an inflammatory response. And this is a self-feeding wheel, and that will ultimately lead to organ failure and death, or can. So we have pancreatic inflammation that causes a systemic inflammatory response in the beginning with an early primary organ failure. And then later on, we have infection and late secondary organ failure. And that again determines late mortality. This is the orchestra. And the slide before that asks you, is this response well understood? And to be frank, I don't think so, that it's well understood. But what this slide should show you that during the last decade or the last 15 years, we have made a lot of progress. And this is when we learned that there's more to pancreatitis than the acinar cell. And I don't think that we have already targeted all compartments, which even in acute pancreatitis are really important for characterizing the disease. So you have innate immunity and you have adaptive immunity. And that is a little bit too preliminary to talk more about that right now. So when we think about the immune response, then we are following a scheme. And that is that of the systemic inflammatory response syndrome and the cytokine storm, hyperinflammation, organ failure. And on the other side, we coined the term of CARS or SARS, and that is a compensatory anti-inflammatory response syndrome or a systemic anti-inflammatory response syndrome. So that is immune paralysis, bacterial translocation, and sepsis. We believe, and this is what we take from sepsis, that these processes are consecutive reactions in the course of the disease. Whether that's true, we don't know. That is just a hypothesis, but I think it is very important to question these consecutive phases if we want to learn how to intervene and how to modulate the immune response in acute pancreatitis. So you've seen, and Anna very nicely pointed out the problem of cell death in acute pancreatitis, and I don't want to go into the mechanism here. I just want to show you that that cell death is closely linked to the immune response. But you will have not only cell death in acinar cells, but you will also have cell death in immune cells. And I will focus on the cell death, which is called pyroptosis, and only a little bit on necroptosis, because in necroptosis, what you actually achieve is the release of pro-inflammatory cytokines and therefore a perpetuation of the immune response. When you look into necroptosis, you know that 40% of cells undergo necroptosis. That is a in pancreatitis, and that is a regulated form of cell death. It's mediated by RIP kinase 1, and you get the MLKL complex. And that is important, it can be inhibited. So if we would come early enough, and that is the major problem, that would be a therapeutic approach. On the other side, we know that macrophages who are phagocytose zymogens can react as dumps and hereby be part of the inflammasome pathway. So we have a very crucial process, which we usually know from acinar cells, and that is that you have trypsinogen released because of the cell death of acinar cell that is engulfed and phagocytosed into the acinar cells. It's fused with the lysosome, and there you have active cathepsin B, and these active cathepsin B cleaves trypsinogen to trypsin, and this trypsin leads to NF-kappa B activation and to the release of TNF-alpha, MCP1, IL-6, so very well-known pro-inflammatory markers early phase or believed to be the early phase of acute pancreatitis, that leads to inflammation and local and systemic damage. So these dumps are important, and they are and those dumps are not only trypsin, but trypsin is probably a very crucial dump in acute pancreatitis, but that can also be bacteria. And they are released from silent macrophages, and the cytokines released will actually determine the fate of the macrophages. So either you will have an IL-6 or IL-1 beta or TNF-alpha IL-12 relief from so-called pro-inflammatory M1 macrophages, or you will have the counter-regulation, so contributing to CARS or SARS, where you need the M2 macrophages. And they are stimulated by IL-4 and IL-13, and these cytokines come from T cells. So there we see we have the orchestra not plays on the adaptive immune system, but also on the innate immune system. And we need both. And we need to understand how they are regulated. So this is to cut a long story short. And it only shows you the process where macrophages are involved. So you have the pro-inflammation, mainly based on the M1 macrophages, again in a self-feeding wheel. So they can be activated by their own cytokine, and then the immune response is propagated. And you have M2 macrophages, where we believe that they probably play a major role in healing. And those M2 macrophages are stimulated by a Th1 and Treg response. And the regulatory T cells, they actually block the pro-inflammatory response mediated by IL-17 or Th17 cells. And they also block the Th1 response, which goes to the M1 response via IL-10. So what you can learn from the slide is that there are multiple factors we need to interfere with to really regulate or modulate the immune response. But what we also understand from this slide is that the immune response in acute pancreatitis resembles some things what you observe in classical wound healing. So if you have a scar in your skin, the same reaction will take place there. So when we actually want to interfere, then we have to interfere not only with SIRS, because that causes hyperinflammation or CARS, but what we probably have to do is we have to address both mechanisms at the same time. So you have to dampen both of the mechanisms. How could you do that? Well, you could again think about pyroptosis and try to regulate the systemic inflammation there. So you have SNF cell damage, you get dumps, you get an NF-kappa B activation, you get the inflammasome reaction inside the cells, then you get an activation of the innate immune system, so macrophage and neutrophils, and at the same time, mediated via IL-18, you get an adaptive immune response with an activation of T cells. So if you dampen the inflammasome, you would modulate the both important players. So that was all true until last year. But now, Aida showed us in a very nice paper doing a site-of analysis in mild and severe acute pancreatitis that there are multiple more macrophage populations characterized by different surface markers. Very hard to say what their actual function is, but that they are involved in determining the severity of acute pancreatitis. So that makes it even more complex to intervene with these with the immune response. So we tried to look and say, what are the major players and what macrophages do we know? And so far I've spoken about M1, M2, but you see there's M2A, M2B, M2C, And we just don't know which of them really contribute at which phase of the disease to necrotizing pancreatitis or mild pancreatitis, or whether they just try to resolve the inflammation. And this is why I think we should definitely go back to Mother Nature and think about, are there other ways? And we have learned from the cancer people, should we not just use our own body to regulate the immune response and just use the self-capacity and renewal capacity of the macrophages? And what it's shown here is there's a transcription factor that is called MUF. There is MUF-C. And so inside the body, a macrophage is able to renew itself through inflammation or stimulation. You could expand that ex vivo, and then you could create builders and killers. And depending on the orchestra of cytokines in your body, these additional amount of cells will actually contribute to a healing process. So we have cell-specific self-renewal capacity of innate immune cells enhancing an autonomous healing and regeneration. So moving from a single target for treatment to modulating the immune response, employing cellular weapons designed and controlled by the host might be another way forward. You could, in addition, also use mesenchymal stem cells. They also have a self-renewing capacity, and we just don't know about neutrophils, but it could be that they also have a self-renewal capacity. We know that they can actually go back from the site of inflammation, go back to the bone marrow, through the lung. Sometimes they stick in the lung and actually cause pulmonary failure in acute pancreatitis. So there could be other processes which we just don't know yet. And therefore, as long as we don't understand the whole process, we should probably ask our own body to help us in modulating the response and not be on the dark side of the immune response like Anna showed us before. I don't know in which pocket the lady should actually put the ball. Thanks for your attention.
Thank you, Dr. Maile. So the next talk is by Dr. Boyan G. And Dr. G is from Mayo Clinic, Florida, and will be speaking on the role of proteases in necrotizing pancreatitis.
Good morning, chairs, ladies and gentlemen. It's great to see you again, although with mask, with face covered. First, I would like to thank the APA organizer to give me this opportunity to discuss about the protease in pancreatitis. I don't know if I can use a point or something. Where's the slide? Okay, can I use that as a talk? Oh, this one. Oh. Okay. As you know, pancreatitis is a common disease in the GI field. I will skip the detail of the disease, but unfortunately, right now, we still don't have a targeted therapy for this terrible disease. So more than 120 years ago, a genius pathologist proposed that pancreatitis is an autodigestive disease. That's what we learned from the textbook, and that's what we are still teaching the medical school students. But in recent years, there's a question that is this concept still true? So I was asked to talk about the protease in pancreatitis. Protease enzyme that hydrolyzes protein to polypeptide or amino acid. Human has about 553 protease. Maybe I can read the name in the next 15 minutes of each one of them. But So they are categorized to several groups by their catalytic residues. But let's group them in another way. So in the disease condition, there's a pathogen protease like bacteria, fungus, and even bacteria like COVID. They use the protease in their own and also in disease. But in human native protease, we can characterize them in non-pancreatic specific protease like lysosome protease, that's in every cell, and also pancreatic protease. That's mainly expressed in the pancreas. As you know, pancreas is a must organ for digestion. You know, because pancreas secrete a lot of digestive enzymes, including protease, lipase, and amylase. For the protease, there are Dr. Whitcomb and their group have a wonderful review. Here it shows pancreas has a trypsin, chymotrypsin, elastase, and several other enzymes. But for pancreatitis, of particular interest is trypsin. Why trypsin? So normally pancreas secretes all those enzymes, but most of them exist in the proenzyme. They don't have activity. Once they're secreted from the pancreas to a duodenum, over there, the enteropeptidase will activate trypsin nogen, the proenzyme. And the active trypsin will further activate more trypsin and other proenzymes. So trypsin is the center of the activation cascade. That's normal physiological conditions. But pathologically, in the Pancreatic-An cells, trypsin nogen can be activated by lysosome cathepsins. And once trypsin nogen is activated in the pancreas, it basically activates the same cascade as in the duodenum. But unfortunately, it will cause pancreatitis because it's a wrong location, wrong time. So over the past 40, 50 years, great progress has been made to study how trypsin nogen is activated in the pancreas. I think everybody in the room involved in this study. But once the proteases are activated, how they cause pancreatitis is less well known until maybe the past 10 years or so. So how to study the role of trypsin in pancreatitis, active trypsin in pancreatitis? There are two ways. One is that you can upregulate the trypsin activity, and another one you can downregulate the trypsin activity. To upregulate the trypsin activity, we can increase the expression of gain of function mutants of trypsin, and including rodent trypsin and human trypsin. And also you can reduce the expression or delete the expression of trypsin inhibitor like SPINK3. So I will go over with you about this. There are several papers published to express the active mutant trypsin. The first one is from Basagi group. They published like using a small rat elastic promoter for pancreatic An specific expression of a mouse PSS1. Actually, it's an equivalent of human PSS1. And they made a mutation that's kind of equivalent to human mutation. They claim they got some phenotype, but unfortunately, it never been validated again. So with craigloxidine, we made a paste trypsin. I'll go over a little bit detail later. And recently, Miklos group has two papers published to mutate the trypsin activity in peptide area, and they cause increased autoactivation. Here's our paste trypsin model. And we started this about 15 years ago when I worked in craigloxidine lab. We have a study to basically study how trypsin and interact with other pathway. And at that time, we use an inhibitor, but we find that the inhibitor is not specific. The trypsin inhibitor can directly inhibit the other pathway, and other pathway inhibitor can also inhibit trypsin. So we thought, okay, can we make a genetic approach to activate trypsin without stimulation? So we came out with the idea to engineer the wild type trypsin by adding the 4 amino acid. That's a PACE enzyme at recognition site. In every cell, in the transgorgy network, there's a PACE. So once the trypsin is activated, the mutant construct will be activated inside the cell without stimulation. So we made a conditional transgenic mouse model with that construct. And surely after induction of the expression with tamoxifen, it causes acute pancreatitis. It's interesting. And all these mice develop two chronic changes with pancreas atrophy, a loss of islet cells, some fibrosis, and mainly fat replacement. So more recently, Miklos group published the two papers by mutant the trypsin activating peptide. One mutation increased 50 times of the auto activation. And when that mutation is knocking into the T7, the major isoform of mice trypsin, and causes spontaneous pancreatitis. Another model, and also they developed chronic changes like the same we found with our pace trypsin model. Another mutation causes only 5 times auto activation, but doesn't cause spontaneous pancreatitis, but it does increase the severity of cerulean-induced pancreatitis. Miklos did a lot of great biochemistry study, and one of them is he showed that mouse trypsin and human trypsin has a lot of differences. So in that consideration, for therapy purpose, the best way is to study human trypsin in the mice. So in that case, the human trypsin transgene will have some advantage. Here I list several human trypsin transgenic mouse models. The first one is from a Liverpool group, and the several others are from our group with Craig Logston, and also 3 of them. And also another one I will briefly mention at the end is a spontaneous pancreatitis model. So this study is from Liverpool group. They use still the rat elastase promoter. It's a small, only 500 base pair. They express the wild type human PSS1 and mutant PSS1, including R122H and NN29. So that's our common mutations in human hereditary pancreatitis. What they found that all this transgene has increased acute pancreatitis model after cerulean induction, but there's not much difference between the wild type and the mutant. So with Craig Logston, we express the PSS1 mutant and wild type by cloning them to the elastase promoter in a bacterial artificial chromosome. So this promoter will be much bigger because it has like a 200 kb construct. So we expect the expression level will be higher than the small elastase promoter, but we still didn't find spontaneous pancreatitis develop. But interestingly, we found that the mutation can interact with some environmental factors to promote the pancreatitis. After that, we thought, okay, trypsin, human trypsin is driven by trypsin promoter. Why everybody uses elastase promoter, because that's, history. So next, we just make another transgenic mouse model with a human bacterial artificial chromosome with full-length trypsin promoter with intron, exon, everything. We thought that will better mimic the human expression condition. But still with this model, the expression is pretty good, but we still didn't find a spontaneous pancreatitis. But after cerulean induction, every mouse gets severe acute pancreatitis. And they all develop to chronic changes with a similar histopathological phenotype as you observed in human hereditary pancreatitis. And also in that model, with experimental therapeutics, we find that actually this trypsin activation interacts with the coagulation pathway. And inhibitors that inhibit both coagulation and trypsin activity has a better outcome compared to either pathway alone. Dr. Pendle briefly already discussed this. I will skip the detail. So in contrast to PSS1, less is known about the PSS2. That's another major isoform in human pancreas. Miklos has some biochemical studies show that PSS2 has a very higher degradation. Then, if it degrades so fast, it may be a protective mechanism because it it's inactivated quick. So, but what's the role in really pancreatitis? It's not very clear. So we made a transgenic mouse model still with a human bacterial artificial chromosome contains PSS2. And we find after cerulean induction, it causes severe acute pancreatitis, and they all developed chronic pancreatitis. So that means the PSS2 is involved the development of pancreatitis. So another way to increase the trypsin activity is to reduce the inhibitor expression. This study is from Japan. They deleted the sphincter 3 in the mice. It's a whole body knockout, and you can find that the Pancreatic-An cell damage starts from embryonic stage and causes the embryonic or perinatal lethality. So trypsin is really important. It can determine the life and death. So another way to study trypsin is to downregulate the trypsin activity. You can use a trypsin inhibitor, but as I said, it's not specific for basic mechanism study. You can delete the cathepsin C, cathepsin that's involved in the trypsin activation, and you can just also directly knock out the trypsin like Ischaemia did. And also you can increase the expression of CTRC. Nicholas did some of this, compare CTRC positive and negative. And also you can increase expression of SPINK3. That's what Roger Little did. Due to time limit, I will just go over briefly some of them. So here are several papers published. So here is a famous study from Ischaemia group. They made a T7 knockout mice. That's a major isoform in the mice trypsin family. And with this knockout, they found that the pathological trypsin activity significantly decreased. And with the decrease the trypsin activity, there are less acinar cell necrosis. But this trypsin deletion doesn't affect much acute inflammation and chronic inflammation. And with the SPINK3 transgene, Roger Little group found that it ameliorated the cerulean-induced acute pancreatitis, and also it rescued the SPINK3 knockout induced lethality. So with all these models, we still don't have a spontaneous pancreatitis model caused by human PSS1 mutation. It's already 25 years after Dr. Whitcomb found that PSS1 mutation is the cause of human hereditary pancreatitis. But fortunately, recently we have a model for this. So basically, we have a transgenic mouse model, and it causes spontaneous pancreatitis in all of the mice at 3 to 4 weeks old. If you want to know more about this model, you can go to the talk on Friday at 5:35 p.m. So let's summary our concept. So protease, including trypsin, is really key in the development of pancreatitis. It may determine the life and death. Lysosomal trypsins are involved trypsin and protease activation, but once they are activated, they will cause pancreatitis. But it will be level dependent, duration dependent, and also location dependent. Dr. Gorelick has a beautiful study basically show once the trypsin is activated, if it's secreted to the duct, doesn't cause pancreatitis. So location is also very important. When we think about the therapy, we should think about not only PSS1 that many people focus on, PSS2 is also important. Not only the mutant is important, wild type trypsin is also important in the pancreatitis. So we also need to think about the human trypsin and mice difference. Their structure, biochemical are very different. If you test your therapy or drug, maybe you'll think about if your drug really target human trypsin. And also other proteins may also be important. So trypsin is really important. Here's a warning. Never say trypsin is not important before Nicholas. A few years ago at this meeting, he said, if you say this, he will fight with you. And also, in addition to trypsin pathway, other pathways are also involved in pancreatitis. Craig Lawson and I have a review about these pathways, and Ishak Solutions study also support this pathway. So even though Ishak is support for the trypsin and expert of the trypsin study, but also never say trypsin is the only pathway beforehand. But to make everybody happy, actually this pathway are interacting with each other. They are not working alone. Trypsin can activate other pathways, and other pathways can also activate trypsin. So here's our acknowledgement. I thank my fellows, they did a great job. And also my collaborators, including Steve Panda, Aurelia, and Richard here, and also my Mayo Clinic colleagues. Thank you very much for your attention.
Thank you, Dr. G. Excellent session on the biology of necrotizing pancreatitis, and we have some time for questions. Dr. Gorley. Yeah.
Steve and Anna, still on?
So Jill and Margie, do we know if our virtual speakers are on?
Yes.
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 They're here. Okay.
First of all, I know that Steve and Anna do not wake up at 5 o'clock in the morning. So I really appreciate the effort they made. So I had a question and a comment. And the first question, I think, goes to both Richard and Anna. And if you think that if we could increase cellular ATP levels early in the course of the disease, and possibly throughout the course of the disease, would we change the natural history of acute pancreatitis and avoid severe disease? And there's reasons I'm asking that question. And I think there's a very important paper from Roger Little's group. I don't think Roger's here, but it was just published. And they looked at the impact of exogenous phosphate in a model of alcoholic pancreatitis and found dramatic effects, improving disease and raising ATP levels. Jerry Schulman's group about 10 years ago looked at the effects of exogenous phosphate on cellular ATP metabolism and found that it could be you could drive ATP levels by giving exogenous phosphate and sodium. It primarily goes in by a sodium phosphate coat. So the question is, and maybe Steve or Beechin knows about this, has anyone looked to see if just serum sodium and phosphate levels predicts disease severity in acute pancreatitis or course? And should we be thinking about the paradigm that that Roger's group studied in alcoholic pancreatitis as possibly more broadly relevant to patients with acute pancreatitis? Because it's certainly, there's many ways to drop plasma phosphate and sodium levels acutely other than alcohol.
Okay, let me start for the first part of Fred's question. We learned in 2000, we think that, if we block necrosis by increasing ATP and block apoptosis by inhibiting caspases, we will get immortal cells. And it appeared absolutely not true. And cell has multiple backups. So organisms have multiple backups to kill cells. And my impression now that really you cannot kill one mechanism and hope to prevent cell death. I'm sure that the cell will come up with something else. Some of program necrosis, they are not associated with significant decrease in ATP, and still they kill it. So I'm not sure that upregulation of ATP only will resolve the question.
Okay.
Okay, can I comment, please? Very briefly, I would say in answer to Fred's question, the answer is yes. And I would say that the critical thing is how it's delivered, and we need to try it out.
This is Steve. I can come in as well. Thanks for the question, Fred. And actually, I was just going to read Roger's paper, and I didn't get to it before this meeting. So, but I'm aware of the concept. So I also agree that yes, increasing the ATP in the cell by mechanisms that it could increase both mitochondrial function and glycolytic function would work. The other thing that that I discussed about the coagulopathy, and also we know that there is decreased circulation, microcirculatory disorders that I think Robert addressed, is that it is probably a delivery system as well, getting nutrients, including glucose and other and oxygen to generate ATP are affected. So I kind of come back to what Anna said, is we have to have therapeutics that address a couple of these things at the same time. But I don't think that's impossible. I think conceptually we can have treatments that address the coagulopathy, the delivery of oxygen and glucose and potentially phosphate too. So I don't think that's too far in the future.
I think there's also worth adding that acute pancreatitis is an evolving disease. It doesn't just happen at once, albeit that whatever it is that's initiated it may be more severe or less severe and determine what is the likely severity without any interaction. But all of the events that have been described in this symposium are not necessarily simultaneous and at one single point in time. They are evolving. And so if we can get in early enough, we can modulate some of these effects. And for sure, we're not just thinking about something that's going to stop the disease. That's way, way beyond any possibility. But something that will ameliorate the disease that is clinically meaningful, that's what we need to aim for.
Thank you.
Great. Thank you, Suhail Hussain from Palo Alto, California. I applaud all the speakers. Several interesting questions came up. Maybe I'll just ask two. And they relate to the proteomics that Steve, you had showed. I guess timing is everything. And Robert, you mentioned it. Often our studies are designed out of expediency, because the samples were obtained when patients were consented for a study and so on. And the proteomic analysis, I would think, is highly dynamic and changes from day to day. And I wonder whether it might be helpful to step back and look at what the course is for any one patient or group of patients in the proteomic analysis for pancreatitis, because I would think that would make a major difference in harmonizing the various findings. And then secondly, related to that, LCMS is obviously standard and helpful, but many groups, including, for example, the UK Biobank, has decided to shift their proteomic analysis towards analyses that capture low abundance proteins. And so using modalities like Olink and so on. And so I thought I'd ask if you could comment on both timing as well as detection.
Thanks, Sohail. First of all, I totally agree with you. This was just one point in time in early disease, and it's both you and Robert point out that disease is dynamic. And you might look at what we did as a pilot to open up maybe some possibilities about exactly what you say. You're exactly right. Also, that the mass spectrometer misses a lot of the lesser abundant proteins, like the cytokines, for example, that you see that we didn't show in the proteomic analysis because it doesn't pick those up. So you have to have complementary methods as you describe, like Olink. I guess the answer is yes, we can do those things. I think from my point of view, we were trying to identify some new observations to help direct us in the future. And so I think that the understanding of coagulopathies in some of these diseases like COVID and potentially in pancreatitis at least makes us think about pathways that could be involved in that. And that's where we were coming from.
Thank you.
Good morning, Steve, Anna, and good afternoon, Robert. Thank you for joining. And I know for Anna and you, it's very, very early, Steve. Great session, great talks. So my question is directed to Steve. Very excellent talk, Steve. I was wondering, when you have a multi-organ failure, and that's what main problem in pancreatitis is, that's why most patients die, and it's a great work. But question is that all these proteomic changes, are they cause or consequence? Are these proteins which are changed? Many of them obviously originate because of initial pancreatic injury and then cause lung injury, for example. So are they coming, so if you want to, based on data, you want to treat pancreatitis, if it's consequence of that, it's already happened, can we make any difference there? So what we really need is what's causing systemic injury so we can make a difference and stop multi-organ. So how would this data take us there? And the second question I had was, again, I think which you have already asked, that is these proteomic changes or any changes in multi-organ failure are very dynamics. Jim Norman, as you might recall, both of us are from that era. There are a lot of these studies which shows that in patients, all these inflammatory markers go up and down in hours, not in days. So, but that's a problem. There's nothing we can do. But if you can comment that is cause or consequence of the proteomic changes of, thank you.
So thank you, Ashok. Yeah, but it is early in Los Angeles, but anyway, so it's still dark here. But your questions are good. So thank you. I think what we tried to do with the Ingenuity pathway analysis is show that there are upstream signals that are originally coming from the pancreas, obviously, that are mediating some of the downstream effects on coagulopathy. So I think that there might be, depending upon the disease state, let's say the coagulopathy has already started, the disease or the treatment might be a little bit different than preventing that from happening. So for example, in the future, I think that we might think at early stage disease, we might want to deal with the first signals, the initial signals like the IL-6 and other pathways I showed that could prevent the whole cascade of coagulopathy from developing. So that might be one approach. But then you're pointing out, oh, let's say we're a couple of days down the line, it's already happened. What do we do? Maybe that's where we deal with the coagulation system, as Bowen did with a prothrombin inhibitor. These are just suggestions. I mean, I think that in some ways, we have to think about how to design experiments that we could start with preclinical models, then move into the patients. But I think you're exactly right. It's going to depend upon where you are in the state of the disease as to what application of therapy that you would use based on these findings. I think the more we learn, the more complicated it gets. But it still points out that we have alternatives for these kinds of therapies. At least we conceptually think about that we could move forward in somehow testing.
Very interesting. I have a question for Julia and panel, who linked, for example, the inflammatory process, central mediators being innate immunity, macrophage, and everything. My question to both of you, and also it may lead to protease community as well, is how much of gut permeability change occurs in pancreatitis? And if so, if there is bacteria coming in, bacteria produce a lot of proteases. Will bacterial proteases could be one of the trigger? It's an open question, please.
So maybe I can answer that, or at least start before Steve can kick in. So bacterial translocation, at least in mice, seems to come from the duodenum. And it's highly regulated by regulatory T cells. That is work done by Matthias Sendler. It's currently, say, in review or submitted. And it clearly shows that if you block out regulatory T cells, then you will actually have a decrease of bacterial translocation. And it's well known that there is ill-perfused, or the small gut is ill-perfused, and that causes opening up of tight junctions and allows bacterial translocation. However, if there would be, so to me, it's a response mediated by the inflammatory system, and it's hardly be, say, influenced by trying to glue it back. You could believe that if you restore the calcium levels, thinking about cell adhesion proteins being highly dependent on calcium, you might actually decrease the gut permeability. But I don't think, at least to the data we have, that doesn't work out nicely. But you would need to address again the inflammatory system to restore gut permeability.
Can bacterial proteases could be another trigger?
For sure. That's
Just maybe, since we covered the inflammation, maybe we need to ask something to protease community also, which we had several experts here. So I was wondering, I think the data is pretty convincing that proteases are activated, whether it's because of trypsin inhibitors not there or whatever. The question really is, at least in my mind, do we still understand that once proteases, whether it's trypsin or carboxypeptidase or whatever is activated, how do they cause damage? How do they really result in overt pancreatitis in the end, which is basically, both inflammation and necrosis or apoptosis. So if we take acinar cells and you put trypsin, would we get apoptosis? Probably not. So how does this happen? Anyone?
okay. I think Ishak Saluja will have the best answer, but he just want to give an exam. I will try. So I think the trypsin activation will cause inflammation or cell death in different ways. Let's say if intracellular trypsin activation, and what we found, it caused mainly apoptosis at the very beginning, at least, and later on, of course, it will cause necrosis. So, but what's in between, it's not very well known. But we all characterize as a kind of model, but the signaling pathway between the cell death and trypsin are not very clear. Apparently, it activated finally the caspase pathway. So another way it causes inflammation, actually, the trypsin is not only digestion, okay? So we have data in our GBC paper with the trypsin. If you add trypsin to the acinar cell, it causes acinar cell damage directly, the blebing, and also it activates the NF-κB pathway, most likely through the PAR receptor, protease activating receptor. So in general, the trypsin, the pancreatitis is autodigestive disease. That's true, but that's not very precisely true. It's except autodigestion, it causes pro-inflammatory signaling pathways. That's, and also except on the acinar cells, it also active the inflammatory cells once infiltrated, and also apparently it affects endocellular cells because the permeability increase. And also apparently it interact with other pathways. For example, like we showed, it interact with the coagulation. And once that happened, it will affect the oxygen supply and mitochondrial dysfunction, all those cascade. So it's not one simple mechanism. It's not only digestion, it's a lot of others. I think Ishaq Solution may have more, maybe I only get 50% of my points, and he has more, Thank you.
All right, that concludes our session. Thank you to all the speakers, especially for our speakers who joined us virtually. We're going to take a little bit of break and come back at 10 o'clock for the next session. Thanks so much.
Bye.